tonight, so please make yourselves comfortable.
This meeting is being recorded.
Got it. There we go. Got it.
Things have changed since you were in Roger.
A rowdy room today. Right. Do feel free to type any questions for our speaker in the chat as we go through the talk, and then I'll endeavour to read them out at the end of the talk, as well as field questions from here in the room. So we've done it before and it's worked okay, so we'll try and replicate that tonight. So just a few words of introduction for tonight's speaker. It's my pleasure to welcome Dr. Tim Ewin, who's based at the Natural History Museum in London and is actually joining us, I think, from Guildford tonight, if I'm right, if I remember correctly.
Correct.
So Tim is online and we're here in person, some of you are online. And Tim has a varied career, having trained in geology at Cardiff and then undertaking postgraduate training and research in palynology at Sheffield and a PhD in paleobotany at Manchester. And he's since worked in the museum sector in a variety of roles, including at Bristol City Museum, and now is a senior curator of invertebrate paleontology at the Natural History Museum in London. And the topic of tonight's talk is on a specialist subject of Tim's, echinoderms, and the talk is crinoids on the national news. And I'll let Tim take over from this point and introduce his talk more fully. So thank you very much indeed. I'll mute myself. If everyone else at home could please mute, we will begin.
Right, hopefully everyone can hear me. And if you've just got a wall of silence, then if someone could just let me know, then that would be great. Thank you very much for inviting me to talk on this subject. It's been a fantastic experience and a real privilege to be involved with the whole project. And I never thought I'd see crinoids, or really any other echinoderm for that matter, mentioned on the national news for good reasons. And it's a relief to say that this is one of those instances. So why have I got that sort of rather sort of gloating title? Is we've got this sort of fantastic specimen here. And you'll see that it's not, from the scale bar at the bottom there, it's not the largest specimen. It's about probably the size of a good sort of serving dish, probably a bit bigger than a dinner plate. But you can see it's got a whole plethora of beautifully preserved echinoderms. These are not normal. It's normally incredibly rare just to find one or two articulated crinoid crowns that you can see sort of with their wavy arms over this block. If you find one or two of those, you're having a good day. And so a block like this, as you can imagine, is really quite exceptional. And this is actually an exceptional block, and I will sort of go through later in the talk as to why it's so exceptional. But it was a stunning thing, incredibly rare, and obviously is packed full of important information about the evolution of this important group of animals. But it's not just one block, it's thousands and tens of thousands of specimens. So we've got a beautifully preserved brittle star here. You can see the mouth and the arms coming off there. The mouth is this star-shaped bit here. You see that on all these arm tips, you can even see there's all the spines that are preserved. So the preservation really is exceptional. We've even got fossil pigment preserved at the site. This is the cup of a crinoid, a Comanche crinoid. I'll explain what these are later. But this pinky colour is actually fossil pigment. And I'll explain a bit more about that. We also have some stunning fossil wood that's got cellular preservation. We've got boatloads of microfossils, and you can see why there's a sort of personal interest in that from my checkered history. And we've also got stunning just preservation of individuals. And this is again another stalkless Comanche crinoid. You can see that all the pinnules are preserved, so on the arms that come off the top here, you see the feeding structures coming off there. There's wonderful specimens, and they are, there's a lot of them in a very small part of the country. So this caught the media's attention as it's really the best UK Jurassic site for fossil echinoderms. We have a very good press team at the Natural History Museum, and I wrote a little sort of press release, and our press team rather sort of shortened it. So instead of saying it was the best Jurassic fossil echinoderm site, it just became one of the best Jurassic fossil sites in the world. And that rather sort of made lots of people sit up. The beautiful preservation of the specimens, and actually I hope to convince you that some of the scientific importance of this material warrants it being of some significance and worth paying attention to. But the splash was well beyond my wildest expectations, and appearing on all these outlets that you can see here. And that was probably in the summer of 21. So about a year and a half ago now, but it was we also appeared on sort of other sort of TV programs here and there, including Blue Peter, which I'm very proud of. I never thought I was going to get my Blue Peter badge, and it was nice to sort of get it maybe 30 years after I was expecting to. So I'll just give a little outline of the talk. Just going to go into the discovery story first, about how this amazing discovery came about, and talk about how we got it out the ground, and then what the geology tells us about the environment, then what are echinoderms, and then give you an overview of the finds, and I'm going to go into some details on the stalkless crinoids, which are focus of the research efforts at the moment. And they're going to end up with a few reconstructions and conclusions. Right, so the story starts with a sort of nice lockdown theme. It seems to be sort of overshadowing everything that we do still. But it was during sort of towards the end of the first COVID lockdown that we had, the restrictions were starting to ease, and we were able to sort of go out and travel a short distance from our houses. And most people sort of went to sort of visit relatives or sort of see sites that they hadn't seen for a while. But two, I call them amateurs, they're really non-professional paleontologists. Neville Hollingworth's got a publishing career longer and more illustrious than mine. But he's not paid for his efforts, and his wife Sally, they sort of have developed a real expertise on looking at fossil sites around Swindon and along the M4 corridor, and they'd seen on Google Maps during their sort of enforced lockdown that a quarry had been expanded on a site that they hadn't visited for a long time. So as soon as they were able to, they got landowner permission to go and look in and explore that quarry. It's very important. All the sites, well, the site that we're talking about is inland on private land. And you need to get the owner's permission to, of course, visit, and also permission to take the fossils there. So we're keeping the site location secret. We're just calling it Wiltshire. That's because it's a sort of ongoing active research site, and we're aware that these fossils are important. They also are pretty, therefore they have a sort of market value, shall we say, and we would rather sort of not have any scientific information lost through sort of unsolicited collecting. We also don't want to cause problems for the landowner. Indeed, we've already had one break-in at the site. If you see any of this material or it's offered to you, then do let me know. But anyway, back to the story. So they went into the site without much expectation. They sort of picked up a few bits and pieces off the floor. They realized they had something important. I hope to be able to show you some of that. So this is the sort of things that when you go to the site, even now, that you can see just washing out. there are these where these orange arrows are pointing to. Okay, I get that they're small, but these are actually complete stalkless crinoid cups. These are normally really rare. And as you can see, there's not just one in the picture, there is a large number of them. And there's actually even more that I could have put an orange arrow next to. I just got a bit bored. There's also other bits and pieces that have come up. You can see there's bits of crinoid, sort of stalked crinoid arms, these longer bits here. And then they sort of dug down a bit, and they sort of came across a layer that was a bit more indurated, harder, cemented. And you could see that there was a suggestion that there was lots going on under the mud. But really, they didn't have any expectation as what they were about to find. And as we can see, there's the same block, and then they took it in, and they've got their own air abrasive unit. You can see it cleaned up beautifully, and there are 1, 2, 3, at least 4 maybe crinoids on this block, and there's actually even an articulated echinoid under there as well. Needless to say, this is not a normal find for the UK or really anywhere in the world for the Jurassic. When you find stuff like that, it's important. So they then got in contact with me on the 30th of November. I cherished that email, and realised it was something important because we've never seen anything quite like that. And comparing it to the material already in the Natural History Museum, and that had been published, it was obviously unprecedented in terms of the diversity and the quality of preservation from a small single site. So annoyingly, that was just before the sort of Christmas was cancelled, and so the second lockdown sort of frustrated visits until really the end of May, when we were able to really travel a reasonable distance and actually go and visit the site, and did a couple of preliminary visits to make sure that there was something there worth taking out. And boy, was there. And we've organised a couple of digs in 2021, and also we've gone back a couple of times this summer as well. The site does flood, so going in the winter is usually requires pumping the site dry, which obviously is not a great use of time. So the people who've been involved, I'm presenting this as a talk by Tim Ewin, but really it's a talk that has been the product of a collaboration of a large number of people. And that's not just people who visited the sites, of which there's been over 25 people, but there's also been, lots of other people have worked on the material, identified the material, shared their expertise about the relevance of the material and the site and the geology of the site. And so it's a big collaborative effort. And indeed, Sally and Neville are remarkable people, and they've been incredibly generous with their time and their expertise, and sharing their finds is something which should be lauded, because they recognize the scientific importance of the site, and they recognize the importance of sharing scientifically important material and not just making money from it or keeping it to themselves. I respect all choices that people make, but I'm just very very thankful and grateful that the attitude that the Hollingworths have shown, fantastic support, and they've always been around to help excavate on numerous different occasions at this site. I mean, we are collecting wonderful fossils, which helps, but it's still greatly appreciated their commitment. So yes, there's a sort of motley crews of various people who've gone down there and dug. I won't go through and name all the names, but how these people and the Hollingworths and myself sort of got it out, I'll just do a quick slide. It is, as you can see, it does flood a bit. So we had to pump that out. The site's actually a wonderful site in that it's not a big quarry. It's probably about the size of 2 tennis courts. But it's the bottom of the quarry is really where the fossils are, and the site is really being quarried for the forest marble, which is the sort of you can just see behind this excavator. There's this sort of brashy limestone. They grub that up and turn it into chippings in a big grinding machine. And then where the excavator's on was Kieran wielding his pickaxe. There's a really muddy, quite a thick muddy layer. That of course clogs up their chipping machine, rock chipping machine, and so they stop. It just so happens that where all the fossil echinoderms are coming out from is that mud and a harder layer just below that mud. So it's an absolutely sort of wonderful situation. The landowners are also incredibly generous in allowing us access and to take ownership of the collections. So I wish I could name them really, because they've been so supportive and generous in allowing us to excavate that. But then, so what we've really been doing is, go through the mud very carefully, lifting these sort of more indurated blocks. As we see in the bottom right there, we got out some pretty big slabs. Then we sort of give the surface scrub, which Dr. Stevens there is doing quite nicely. And that just sort of takes off the mud layer and reveals whether we've got articulated echinoderms there or not. And by and large, we have. And then she didn't want to have photograph, but Sue there's is collecting some clay for bulk sampling, and there's some quite interesting results from that bulk sampling as well. So that's a really nice thing. So back at the museum, so we've sort of labelled it all up. It comes back in these boxes, all wrapped in bubble wrap. Then we have to put it through quarantine, and that picture that you can see there is about 1/3 of the material we've collected. So we've probably collected over 300 individual blocks, all of which have got echinoderm material on. And then it goes through to the conservation and preparation team, and you can see Mark Graham here in the bottom right, using an air abrasive to prepare the surface. And you can see in the top right there, there's a beautiful stalkless crinoid coming out. So I've got a little video here of air abrasion. So the other wonderful thing about the quarry is that it's in fairly soft clay, and so it's actually really very easy to prepare. So I've got this nice video that you can see gradually the clays being sort of blasted off by this miniature sand blaster, and you can see the crinoid arms starting to appear underneath that. Those little black specks are actually bits of fossil wood. There's no shortage of it, so we don't mind losing some of that, but we have kept samples of all of these things for reference in the future. And you can see it's really sort of coming out very quickly and easily. This is not normal. I should hasten to add. It takes a lot longer, but because the material is so easy to prepare, that we can actually prepare large numbers, which again helps with identifications and also looking at variation between individuals, which is something that you can only do at a handful of sites across the world. The other nice thing about this video, which I will speed up a little bit, is that you can see that it's actually 3-dimensional. And it'll turn it over and it'll blast away at the other side. And you've got this beautiful 3-dimensional crown, almost as though the animal's alive and had just shed its crown shortly before being dug up by one of our colleagues. So yeah, nice one. The sieving, which I showed Sue was doing some bulk sampling, was done by David and Alison Ward, who again are scientific associates of the Natural History Museum, not paid paleontologists, but are experts mostly on fossil fish in their own rights. But they also have done a lot of work on microvertebrates, and in their quest to find some early mammal, dinosaur, and reptile teeth, they sieved about 1.5 tons of the clay forest. And that resulted in this rather sort of jumbled mess that you can see here on the left as I'm looking at it. The one without the badge, that is actually about 3,000 individual stalkless crinoids, which is an unprecedented number from any site in the world. We've also got perhaps over 150 stalked crinoid crowns. These are isocrinoid crowns, which is behind the badge, you can see there. So these are large numbers. And I greatly appreciated to David Ward, because otherwise a lot of that work would not have been done. We've got a lot of bulk material that volunteers are picking through at the moment to ensure that we get as many different taxa as possible. So what does the geology look like? So we're in the Bathonian here. So this is the part of the middle Jurassic. You see a nice big red arrow or arrows. So we're in the forest marble formation. The forest marble is a very variable formation. I'll come on to that in a second. But we're in the Cladoniaceras discus zone for those that are interested. So it's part of the Great Oolite group. So you've got some Bath stone just below us, and a little bit of the Cornbresh above. So it's an interesting dynamic system. What did the world look like 167 million years ago? Britain was sort of located in between two sort of larger continents of Fennoscania and Laurentia, with the Tethys Ocean to the south, and there was a Boreal Ocean to the north. And this was just as the Proto-Atlantic was beginning to form, and Britain was right on that plate margin at the time. And most of England was underwater, and there was a few areas of high, which is clearly demonstrated by these yellows, and then a little bit more detail where the site was sort of towards the this rather sort of nebulous tip of the Anglo-Brabant landmass, and that's quite you'll see the relevance of that in a minute, because the site is marine, but it's not very far away from land. So what did the middle Jurassic look like? Of course, there were the oceans had ammonites and a few other invertebrates, and then there was things like ichthyosaurs and plesiosaurs, belemnites. Interestingly, at our site, we don't have any. We have very little nekton at all. It is almost all invertebrates. We've got a couple of fish teeth, like literally 10, 15. Couple of shark's teeth, but we have no marine reptile remains. We don't have any ammonites either. There's potential that it's to do with taphonomy, but we think it's actually sort of probably something to do with the actual sort of burial history at the site. On land, of course, we had this is the very similar age to the Yorkshire Jurassic flora, which is incredibly rich. Over 150 species are known from those beds, and you can imagine that there was similar vegetation on the London Brabant landmass as well. It's mostly conifers and cycad-like plants and ferns and bits and pieces like that, even the odd club moss and horsetail. So yes, very different today. Very no angiosperms, no grass or anything else like that. And of course, there were these sort of boring, sort of large terrestrial reptiles that people are interested in, called dinosaurs. Again, no dinosaurs at our site, but it was right slap bang in the time when things like Camarasaurus are found in quarries nearby, and stegosaurs as well from terrestrial sites just outside of Swindon. And you can see some of those on display at the Natural History Museum. So site stratigraphy, as I alluded to earlier in the talk, the fossils were all found from the quarry floor. You can see these little sort of star-shaped things in this rather detailed log, which was created by Professor Charles Underwood at UCL very kindly. And there are the odd echinoid test that's found in the sediment above, but really the rare and the exceptionally preserved numerous echinoderms are all found at the base of this quarry in 2 layers. And that's pretty much the case across the whole quarry floor, although it is incredibly difficult to trace one bed from one part of the quarry to the next. There's a lot of lateral variation, and there's a lot of thickness variation, and some of the units that are hard in one part of the quarry just form almost a continuous bed of clay in another. So it's a really diverse and dynamic site. It's very frustrating because we were hoping it was going to be a lot more straightforward, but I think it is going to turn out to be fairly straightforward. We just can't trace things as well as we would have liked. So there is a lot of what does the sediment tell us? It tells us that it's an open marine nearshore environment with terrestrial and freshwater input. So it's got to be open marine, because otherwise we wouldn't have a large number of well-preserved echinoderms there. But we've got evidence from the palynology, from ostracods, and enormous amounts of fossil wood that there is evidence that there is a lot of different input into this site. And on top of that, we also even have radiolaria, which are incredibly rare in the UK Jurassic. So there seems to even be an oceanic input, because radiolaria are only found in oceanic waters. So it appears that there is obviously several currents or sources of nutrients, shall we say, that are all converging on this site. This is probably an explanation as to why there is such a high abundance of echinoderms. You see the specimen there on the left of the screen as I look at it. You know, it's just a lot of echinoderms, and they're all on top of each other. They haven't travelled very far. So the sort of environment that we think we're in, there's obviously a lot of freshwater input. So we've evoked you know, a delta of some sort. We're very much not in the delta, because the sediments are all sort of shelly sandy limestones. Shelly sandstones, and overlying the sort of finer muds, and those muds can occur in different parts of a delta front. So they can be just off the front, they could be just off the sandbars, or they could be in slightly deeper water. But we're not talking hundreds of miles from land. We're talking, you know, 1s to 10s tops. So it's a sort of interesting dynamic area. And of course, as the delta has prograded basinward, then all of these sandy units will then start to cover over the mud, which is what we're seeing, which is what's capping it and preserving the fossils at the site. So I've mentioned a lot about echinoderms. Just to give you a quick whirlwind what they are. So echinoderms are a wonderful group of very alien looking organisms, incredibly diverse. There's over 7,000 living species. They have a wide range of ecological habitats, a wide range of behaviors, everything from suspension feeding to active predators, to sediment filterers, to grazers and all different shapes and sizes and different ways of doing those things. They are very charismatic. Some lots of crinoids, modern crinoids move, crawl around. Brittle stars, wonderful creatures, very active and mobile and very quick. Starfish, less so, but have sort of interesting quirks, like they can push their stomachs out through their mouths and feed outside of the body, which is quite amazing. And then this is my daughter here on the right, and she's holding a nice Asterias rubens, or common starfish, on a beach in South Wales. And this is to remind me that echinoderms are also important from the perspective is that they're deuterostomes. So in the major division of the animals, chordates and craniates and vertebrates, and you and I are deuterostomes along with our echinoderm cousins. So we share a common ancestry that's closer than we are to things like insects and crustaceans and snails and octopus and things like that. So they have a direct relevance to us as well, although they've diverged quite spectacularly from us. So the key things about an echinoderm is that they have fivefold symmetry. At least all the modern ones do. Those in the Paleozoic are very different, but I'll leave that for another talk. They have a hydrovascular system, which they take in seawater, they pump it around a series of canals, and that fires their tube feet. And they also have a skeleton that's composed of, and those tube feet are used for a wide range of things. In starfish, they're mostly used for movement and also prizing open mussels or other bivalve shellfish. But they can also they do feed in other ways. Other echinoderms use their tube feet for catching suspended food particles or for burrowing and things like that. So yeah, dynamic animals. Then they have a mesodermal skeleton composed of calcite plates, which are often referred to as ossicles, and these have a sort of sponge-like galleried structure which we call stereome, which is sort of 50% calcium carbonate skeleton, but it's also 50% soft tissue in life. So it's a very characteristic thing, almost all modern echinoderms share. So they're totally marine. They have amazing powers of regeneration. You can see here on the top right that everyone knows that if you pull an arm off a starfish, the starfish can just regenerate that arm. But what not so many people know is that in some groups of starfish, in this instance, you can actually pull the arm off, and the arm that you've pulled off will grow 4 small arms and completely regenerate to an adult form. Not all starfish can do that, but some of these sort of ones with longer arms can. So yeah, be careful when you're pulling starfish apart. Yeah, wide variety of ecological strategies, as I just said. And you can see down here, this is the extraoral feeding of these starfish, so that they can actually push their stomachs out, and they'll push the wall of the stomach up against a large food source, in this case a dead sea lion, and they'll get nutrients out of it that way. So yeah, they're quite incredible creatures. They are rare as fossils because the plates that have been describing are held together by soft tissue. Whilst they might feel hard when you pick them up, or that plenty of people have picked up a dried starfish in the past and they are quite solid, they're a bit like like a sort of human skull, as it were. It's sort of hard to the touch, but there's a layer of soft tissue between the bone in the outside, quite thin in my examples. So yeah, most they were particularly affected by the mass extinction of the Permo-Triassic boundary, and most of the groups the orders that we see alive today, particularly in crinoids and in starfish and even ophiuroids and echinoids, can really trace their origins only back to the sort of lower Jurassic, and so their appearance obviously occurred sometime during the Triassic. But most of the modern groups have a fossil record that goes back to the Ordovician. There's no crown group echinoderms known from the Cambrian, which is interesting and the subject of another talk. So yes, Middle Jurassic echinoderms of Britain are very well known. Also up until the discovery of this site. And what's interesting is that this site includes a large number of taxa that have not been described from Britain before, but have been described from elsewhere in the world, particularly in the Swiss Jura, which is interesting. And I think it reflects the very dynamic nature of the depositional setting during the Bathonian in the UK, where you had a sort of patchwork of tropical, subtropical environments, lagoons, deltas, sandbars, etc., etc., etc. And so there was obviously a wide variety of different echinoderms which are specialised there. So some of the classic examples you see was Apiocrinites, Parkinsoni / elegans, which is known from a very famous spot in Bradford on Avon. But there's also other examples, Alsacrinus down there. So just to give you an overview of what we found. So I hopefully just sort of wow you with some pretty fossils, if nothing else. So returning back to this block, which I pointed out to at the start, so we got nice stalked crinoids. This is Isocrinus Andre. This is a special block because it has more than just isocrinid crinoids on it. And you can see that there's lots of them. I've never actually counted them. There must be at least 20, 25 individuals on this block. But those with good eyesight, in the top right there's a nice circular organism there with some spines on it. That's of course a sea urchin. It's probably Perisolania. Sorry, it is Perisolania. Then down a little bit on the same side, you can see this sort of triangular shape. This is actually the arm of a starfish. Mastropectinid, beautifully preserved with spines and everything. Then here is very difficult to see until it's pointed out to you that this is the disc of a brittle star. And you can see the arms coming off here. And those with exceptionally good eyesight, there's actually a little cluster of plates around here. We can see these little spines on it. I hope that's clear to you in the hall. And if it isn't, you'll just have to take my word for it, that these are actually the mouth parts of a sea cucumber. So on one block that's maybe the size of a large dinner plate or serving plate, we've actually got all 5 classes of extant echinoderms represented on a single block. That is rare. Probably only known from maybe 5 or 6 other specimens in the world from any stratigraphic horizon. So it's exceptional stuff in terms of its diversity, and having all 5 alive at this one site and being preserved shows how exceptional it is. Although you can go to many modern sites and you can find all 5 classes living side by side, They just don't often preserve together like that. And that's an interesting observation in itself. So other things I've already suggested about showing some echinoids, but we've got the large primary spines, but you can also see there's sort of hair-like secondary ossicles as well. And we've got 5 different taxa. There's a wonderful Stromachinus here. Again, you can see all the very fine hair-like spines on it. This was an organism that probably lived sort of either informally or semi-informally, so it's sort of burrowing through the sediments, whereas these Paracelanians with these very handsome, large, long, robust spines, and they have very strong jaw elements. They were potentially feeding either on other echinoderms, and there's a suggestion that they could be predating the stalked crinoids. Sorry, the crinoids, or they could be just processing algae. It's difficult to say conclusively one way or another, but they are there in large numbers, and they're beautifully preserved. Also, the astrozoans, so the starfish and the brittle stars, are there in profusion, and the quality of preservation, as I've said before, is exceptional. We've probably got a new species of brittle star and probably 2 or 3 new species of fossil starfish. Their preservation, 3-dimensional. These things have undergone a little bit of transportation, but it's sort of probably meters, not miles. Otherwise, they just break up. Yeah, so it's wonderful stuff. Plenty to be getting on with, to describe and compare. I mean, the starfish on the left there, as I look at it, is absolutely exceptional. You can see that it was buried underneath all of this sort of crinoid-rich mud. There's actually a second starfish next to it. And then this whole thing's upside down, if you see what I mean, and this layer where my hand is, as you can see, I've turned the image around here. That's the way up it was in life, and it's so exceptionally preserved that you can see that the animal was obviously alive when it was buried, and you can see that the sediment has come from the left to the right, and it's actually flicked the tip of the starfish arm up, which is just, yeah, like you're capturing a moment in history. So this is very clear evidence that these animals were living as a community together. They were buried very rapidly in quite a catastrophic event, and then a short time later it was established, and then they were buried again. And I'm able to say that because very clearly we've got two distinct beds with articulated echinoderms in them at the bottom of this quarry. And the specimen on the right here, you can see there's other wonderfully sort of aligned crinoid, complete crinoids, and also the swept arms of this other starfish. That was a wonderful find. That was actually completely buried when we first started preparing it. We didn't know it was there, so it sort of appeared beautifully. There's actually another brittle star just there. Yeah, so lots of stalked crinoids, and you can see by this specimen that they have really nice long stems, which reminds me to say that we've actually got a research grant looking at tiering in the fossil record, and these are going to give us an indication of how structured the plankton suspended food particle feeding organisms at this site were, and comparing that to modern sites, and also sites of similar crinoids from the Paleozoic, to see how these important communities evolved. And then we've got a boatload of these beautiful stalkless crinoids, or comatulids. And these are proving to be really interesting as well from an evolutionary perspective. And I will go into what crinoids are and what comachlids are in a little bit more detail to give you some, to contextualize why it's important. So hopefully that won't take you too much time. But again, just beautiful, beautiful, beautiful material. Yes, you can see they're alive today. Indeed, they're the actual, they're the most diverse crinoid, and at the side they are wonderfully diverse. And just to quickly pop these last two slides up before I dive into crinoids, is the picture with the drawers shows the total holdings of the NHM collections, which represents about 200 years worth of collecting. And we had about 25 to 30 individuals. And just for David Ward's sieving efforts on the other side there, with this big jumble of stuff here, we've probably got about 3,000 comachlids. So it just goes to show we've got sort of several orders of magnitude more material than we did prior to finding this site, which is utterly phenomenal. And really it's phenomenal across the world. There is no other site that really compares from this age for stalkless crinoids. And then just to really, we've got the disarticulated bits. We've also got the articulated bits. This is a big block. You see it's quite a reasonable size, probably the size of a sort of small desktop. And you can see that there's a suggestion of, there's a couple of bits and pieces you can see poking out there. Actually, as you look more and more, there's lots of little lines and squiggles and wiggles. And I've circled each individual comachlid on this surface. And if you don't want to count all those red rings, there's actually 47 individuals that are articulated on this one bedding plane. So these animals were living at this site in huge, vast numbers and very dense. Right, and that's quite similar to what we can see in parts. So I'm just going to introduce crinoids now. So most people are familiar with the stalked crinoids. You're often taught that they are permanently attached to the substrate, which was true for about 50% of the Paleozoic taxa. But even the Paleozoics and most of the Mesozoic ones can actually move. So if you got an idle moment, if you type into YouTube crawling crinoid, you'll see one of these neocrinus crinoids dragging its stem along by using its arms. And this is part of why they're deemed to be successful in that they can escape predation. And then on the right, there's this Proisocrinus from the Central Pacific. This is actually a cementing variety. So they put these arms up and they catch suspended food particles. And they sort of can aggregate together. So they have larvae, which helps with their dispersal. They can grow fairly quickly. They grow up to this size in 3-4 years, sometimes quicker, sometimes slower. Yeah, they're very interesting dynamic animals. And then what we've got here is you've got a nice sort of community with some stalked crinoids at the bottom, and we've got some stalkless crinoids at the top. These nice little yellow cirri. So these sort of leg-like things that are coming off the main stalk here, they're used for attachment. And they can actually relax the stem, and that's what sort of can form a curve in it, and then they can make it rigid again. They have a very interesting material called oh gosh, the name now. Anyway, mutable collagenous tissue. There we go, which can act a bit like a muscle, but it's in addition to muscles as well. That can stiffen and relax the stem, and they don't expend any energy when they're stiffening that mutable collagenous tissue. So they're able to hold these feeding postures for a long old time. Right, and then oh, I've pressed the button twice, but this is the stalkless crinoids, and many of the stalkless crinoids can actually swim, as this one's doing in a little tub in a research station in Japan that I visited in 2019. So they don't just sit around and wait for the world to happen to them. They can move around, and you can see here the stem is being reduced to a single compound ossicle, and they have all these little sort of leg-like cirri that come off that they use to hold on to the sea floor or some sort of substrate, in this case coral, and they use the arms to catch suspended food particles. These the stalkless crinoids in the modern environment, the only ones that are found in the photic zone in any sea in the world. The stalked crinoids are restricted to deeper and darker waters, and that's probably due to the evolution of Juraphagous fish, so fish that can crunch up hard calcitic skeletons. But because these guys can swim and they can move, they can climb and crawl, it means that they can retire into nooks and crevices within the reef head during the day, and then they come out at sort of dusk and disappear at dawn to feed. So yes, dynamic little creatures, and very interesting. And they can form, as is probably quite obvious, these large meadows, as they've been called, or submarine animal forests. A bit like an animal forest, you can see that there's the stalkless ones here and the stalked ones there, and you see that there's actually a couple of little brittle stars dotted around as well, which also form part of this the canopy of the forest, this submarine animal forest, as it were. So they have a long fossil history, and the modern groups are really confusing, so that there are some that are stalked well, there's several different unstalked crinoids that have given rise to stalked groups. So you'll see within this clade of stalkless crinoids, there's this group of stalked crinoids, and then there's also independently some of these other groups throughout the fossil record have actually given rise to stalkless crinoids. But the ones we're really talking about are the camachlids, which is almost the most crown of the of crinoids. But of course, what we're looking at in the modern record is really a vestige of what we have in the fossil record. So there's a large number of different groups, and we've only really very recently, Selena Cole and Davy Wright, along with their collaborators Bill Osich and a few others, have got a grasp on how Paleozoic crinoids are related to each other. And sort of just to sort of give you an idea of what's been lost, you can see that all this diversity in the Paleozoic was lost, and then a single lineage gave rise to what we can see in the modern environment. So they're an interesting group that were hit hard by the Permo-Triassic extinction, but recovered quite spectacularly. But those that recovered spectacularly all have a common ancestor. And on top of that, you can see that they had a larger diversity in the Paleozoic, very famously, until really the present day. And the present day is really illustrated by camachlids. So you can see this nice diagram here that Janevsky produced in 2011 for his PhD thesis. So this is Mesozoic, this is Triassic here. There were no known camachlid crinoids, and they appeared probably towards the end of the Triassic, lower Jurassic at latest. But in the modern environment, there's over 570 of the 650 crinoid species are camachlids. So they are incredibly important in modern environments. but they're rare in the fossil record, particularly in the middle and lower Jurassic. And they're only really known larger numbers from the lower Cretaceous onwards. So we're sort of talking from about here-ish up. And that's important because what we're finding at the Bathonian site is this Andymetra. And that's important because in the Jurassic, really, there's only 3 genera well known, which is the Crinoids, the Paleochemaster, and the Andymetra. So we've got one of the 3 most sort of, well, of the oldest, one of the 3 of the oldest Camachlids in the fossil record, which is interesting in itself. Although there are, they all belong to the Paleochemaster, but they're a lot older. So we're looking at this, what appears to be sort of the start of a sort of diversification event, but whether that's true or not, which it probably isn't, it's probably an artifact of the fossil record, it still provides an interesting view into the evolution of these creatures. So a wonderful paper by Hess charting the early history of Camachlids, identifying this sort of radiation with Paleochemaster and Paracomatula coming off a pentacrinid, which is a stalked crinoid ancestor. So Pentacrinus is, very famous, is the one that you see that comes out a lot of Lyme Regis, and also these sort of huge sort of log-like arc things that you get from the Holzmaden of Germany. Look those up if you're interested. But recent phylogenies using molecules and morphology has suggested that some of these, actually all of these early crinoids are actually early derived. You can see there's a Solanochronites here. You see Solanochronites is right up in the crown. Whether that's its true position, I'm sure some refinement is needed, but this does appear to be a very derived Camachlid. And the reason why we think it's a very derived Camachlid is that we've been able to look at the multiple different characters of fossil and modern Camachlids, or Salisbury and Zamorodid. They've looked at the body cavity, and you see that the body cavity is not just a simple cup, but there are actually little canals which go between the plates where the ossicles join, and the ossicle margins, and actually into the ossicles themselves. You have these excavations where the body cavity extended into. This is thought to help with sealant circulation. But you'll see that the structure is actually quite consistent in the different groups. You can see here there's not much elaboration. There's maybe 1 or 2 little sort of finger-like projections in the more basal taxa. And then as you go higher up, more derived, you've got a greater elaboration. And you can see in this later paper by Salisbury that they got really very, very interesting indeed. So that is important for us because we're able to actually scan the Camachlids in a CT scanner, micro CT scanner that we have at the Natural History Museum. And we're able to look at the density contrasts in that, and we're able to identify the body cavity, but also it's the cavities that are between the skeleton. So, and then we're able to colour those density differences, which gives us a very nice idea of how that looked, and then we're able then to compare that to modern the structures in modern crinoids. I don't know if we can. Oh, okay, we're going to do that again. So the green is the body cavity, which is unhelpful in that it's red in the Salisbury and Zamora paper, and the red in the ones on the right is actually nervous tissue. So you can see that the structures are beautiful. You also see that they're actually quite distinct, and they're not like really the ones that are depicted there, which is interesting. Okay, so I thought I had some more slides in there, but obviously they didn't make it. That is interesting because we're able to also scan some other. We've also scanned some other Middle Jurassic camachid central dorsal. So we've actually managed to look at cellular crinoids, which if I go back, hopefully, yeah, cellular crinoids. So which is this taxa here, fairly high up, but and we've actually also been able to get a paleocremaster. So if we go all the way back to our friend Hess's construction, we've got all three groups from the Bathonian in the Natural History Museum collections, and we're knocking up CT scans of those. We've currently done selenocrinitis and Andymetra, and we're just starting on a paleocremaster, and all three appear to have very different structures of their body cavities. So here's some pigment I was talking about at the start. This is actually fossilized pigment. So when people tell you color doesn't preserve, they don't know crinoids. So this is actually, I'll read it out, a bioactive polycyclic quinone pigment related to hypericin. That's been established using a variety of geochemical techniques, largely based out of Klaus Wolkenstein's lab in Göttingen, Germany, and is collaborating with us. And this is apparently some of the oldest example of fossil color in camachlids, which is a very nice thing to be able to say. This is not to say that all the stalkless crinoids at the site were actually pink or purple. It's more to say that this quinone pigments are actually, as far as I understand it, are to do with the sort of metabolic pathways, and they sort of help with this sort of transfer of electrons. So a little bit like our blood is red, not because, and it doesn't mean to say that we're completely red. It's just that the pigment is red because of its structure, and it's that structure because it carries out a function. And it's the same with these crinoids. They probably weren't purple. They could have been a variety of different colours, and if they're anything like the modern ones, they would have been fairly brightly coloured, but they nevertheless have this pigment preserved. We also have an apparent partitioning of crinoids at the site. So because it's a floor, and we're able to sort of compare one horizon in one part of the quarry to another. I know it hasn't done that. We're able to actually identify that the lower layer in the sort of bottom part of this quarry here, it's been almost all stalked crinoids, with the occasional like one or two stalkless crinoids. And then 4 and 5 and up towards this area, the north of the quarry, it's almost exclusively stalkless crinoids. So a bit like in modern environments, sometimes you get they're living side by side, but they are often not as integrated as you would have thought, and they sort of they're adjacent to each other, but they're still quite distinct, which is quite an interesting observation that we're able to make at the quarry. And you'll see that the quarry is not even 30 meters across, and so we're here talking about maybe 5-6 meters. You go within about 1 or 2 meters, you go from being almost, well, predominantly stalked crinoids to predominantly stalkless crinoids. Very, very, very interesting. We've also got a whole plethora of other shelly fossils at the site, brachiopods, bryozoa, some nice encrusting circulids. So obviously this stuff wasn't just alive and then was buried. There was obviously a lot of these skeletal elements were lying on the sea floor and probably were brought in during a sort of submarine mudslide. So there's lots of oysters as well, and bivalves with a calcitic shell. I'm sort of emphasizing that because there might be a taphonomic things. There might be very little aragonite being preserved at this site. But we think because of the nature of the burial and it's instantaneous, then unless there was going to be lots of dead ammonite shells lying on the sea floor, then they wouldn't have necessarily have been caught up in the depositional event, and therefore being able to swim one way, very similar to the fish. But why the fish teeth are not being found at the site, and why there is no dinosaur teeth or terrestrial animals that are being washed in, particularly as there's so much terrestrial wood there, is somewhat of a sort of enigma to the site. But we've also really interestingly got a wonderful fossil crab, and we've got several crab claws, maybe 15, 20 crab claws now. So there were obviously lots of crustacean living there as well. So yeah, quite a dynamic community, not just echinoderms, but echinoderms with a pretty things. So just to sort of pile that in, the fossil history of Comanche meadows is that they were unknown prior to the Lower Cretaceous. So the occurrence that we have pushes that sort of type of environment back 20 million years, which is no mean feat. So an important snapshot on the structures and interactions of those creatures. And indeed, combined Comanche stalk crinoid marine animal forests are now only found in the deep water environments. Comanche is still found in shallow water environments, but the stalk ones are, and we think our site is probably well within the photic zone, considering everything that's there and its location. So we feel that it's important that these animal forests are important biodiversity and biomass hotspots. And so it's important to understand their origins in the modern world, and this appears to be one of the earliest instances of this type of marine animal forest. So yeah, so the BBC did this very nice little reconstruction for us. So this is all of us working the quarry, and then it's going to sort of go into a sort of reconstruction of what it was like. So 167 million years ago, okay, we probably weren't that close to land, but there was a delta nearby, and then under the water there was this wonderful, rich ecosystem with starfish, sea urchins, crinoids, all the odd sea cucumber, all living happily together before they were buried by a submarine mudslide. What triggered the mudslide, we don't know, but it's a very dynamic setting. So you can imagine build-up of sediment from a delta. There's a large storm or an earthquake and dislodges the sediment, and down it comes and buries all these creatures. So, like I said, this is a large number of people that have been involved in the project, and it's not just other academics, but it's other people who've helped with collecting, people help with sieving, people have helped with processing all sorts of data for us, and even the media teams and the press teams have all been fantastic, and fantastic support from admin staff as well, getting all the health and safety sorted out. So brush over that and say thank you very much for listening, and hopefully I can take some questions if anyone's got them. I don't know what to do, so thank you for listening.
Wonderful. Thank you very much indeed, Tim. I don't know if you heard the round of applause coming from the room or not, but we were certainly applauding your talk. Thank you. That was lovely. You know, absolutely staggering images, not only of the fossils as we were hoping, but of the modern relatives as well. That was really, really great. I think a lot of us remember seeing the headlines, so it was great to find out more about the site. Can I take any questions? Let's have a look. One from Roy in the room.
I was interested, we seem to be leaning towards interpreting the material as essentially life assemblages. And yet, by looking at it, it didn't seem to be that at all. It looked more like a catastrophic death assemblage of stuff that's been brought in some considerable distance and piled up on top of one another and sorted, very much like a storm debris on a beach or something like that. I mean, it's not an unusual sort of situation, but none of the fossils, or few of the fossils, were actually complete. They were mostly large chunks of nicely articulated fossil, but not complete. And you mentioned also the variety of other materials that are associated with the deposit. It seems to be more that it's allophilous material rather than autophilous, I suppose. Do you care to comment?
Thanks, Roy. Now, I don't know if the microphone picked that up for you, Tim, or people at home. So, but I'll try and paraphrase. Did you get that clearly, or I can paraphrase?
I got it, and I understood the point.
Great. Yeah.
That was being made. There's distinctly two different types of shelly fossils there. There's the well-preserved para-autochthonous material, and there's material that is encrusted and broken up and obviously has been brought in from a more reasonable distance. Things like camachilids do not travel very far and are preserved in that state. Their arms fall off and you end up with very little other than the central dorsals preserved. And a lot of the sieved central dorsals, we've actually CT scanned a lump of the sediment that they were coming out from, and they are all articulated in there. It's just the sieving process has actually broken them up. So I take the point that there will be some grading in there, and there will be some selection, but I think with a lot of the crinoid material, it's very difficult to see that happening in a big way, or happening a lot before they're preserved, before they're buried, and that sorting process ends. There wouldn't be much time for it to do. You could have a storm event where the thing's just being turned over. That's the sort of amount of movement you're thinking, or that you have this sort of sudden influx of material, and that's burying, moving the specimen slightly, but not. We're talking about small numbers of meters, not hundreds of meters or miles. But some material has. And we can see it's all broken up.
Thank you very much. Any other questions? Another one in the room here.
I was interested to hear about the preservation of colour in some of the specimens, and I'm wondering if there's the remotest association with the celebrated colour preservation in the sponge, I think the sponge fossil in the Middle Jurassic in the Cotswolds, the Selenophora fossils found. Am I right? Is it sponge?
Red algae.
Algae, sorry, red algae. Thank you.
Yeah, I think it was originally described as a sponge, wasn't it? It was a beetroot stone or whatever it's called.
That's what I'm referring to, yes.
Yeah. I don't know off the top of my head what that colour has come from, but I do seem to remember it was also a pigment of some description. I think it would be surprising if it was the same particular group of pigments, but I don't know enough. Thank you. I will look into that.
Thank
but
you.
just to say a little bit more that that crinoid pigment is well known throughout the fossil record, and actually people have discovered that purple pigment going right the way back to Paleozoic crinoids. I think it's to the Ordovician, to the upper Ordovician parts of America, and these weird purpley colours to the crinoids. So the fact that we've got it in the Comanche Desert, the Wiltshire site, is not that surprising in that context, but still, it's nice to have the oldest one in Comanche.
Oh yes, in the room again. Thank you.
Yeah, this is related to Roy's question, I think. It seems to me that the mud that came in is absolutely critical to the preservation of these fossils. But I'm not entirely sure why it has to be to do with a delta. You can get plenty of muds on continental shelves, nowhere near a delta. And sure, all you need to do is to move it in some storm event and dump it on top of your unfortunate crinoids.
Absolutely, good point. I've rather abbreviated that part of this the presentation, but we've had a large, we've had the nanofossils analysed, the dinoflagellates and palynology analysed, and the ostracods analysed, and they all say the same thing, which is there is a terrestrial and nearshore input. And combine that with large amounts of well-preserved fossil wood. We've got some logs that are over sort of 2.5 meters long, all the way down to sort of little granular sort of bits. I think the most compelling evidence is probably from the ostracods and the palynology, that it's near shore. But there's obviously a wide variety of organic input from lots of different environments, and so there seems to be maybe some form of ocean current or marine current that's bringing in nutrients, as well as a lot of material coming in from a terrestrial environment. And just looking at the paleo geographical reconstruction, land was nearby. So we're not talking about a sort of Amazon River input, but we're talking perhaps something smaller. And so it just seems to all make sense with the microfossils and the paleobotany. We've got cellular preserved leaves and all the rest of it. It's a nice suite. I guess the thing to do would be to publish all these results so that people can criticize and make I can share all the data.
Thanks, Tim. I can see a question has come in from Mark Evans in the chat. And Mark asks about the diversity curve. And is it the case that very few, maybe only one lineage of Comanche make it through the KPG boundary?
No, the crinoids, that type of crinoid, the Comanche seem to do okay. I think there was probably a different story for things like Rovia crinids. It appeared that they were diversifying in the Jurassic, and the evidence that we have from the sort of molecular and morphological phylogenies that have just been published in the last 2-3 years, it really suggests that there was a lot of diversity that has not been recognized in Jurassic fossil record. And that's probably one of those problems of the fossil record. Although it seems that, or maybe a lot of this diversity was happening in parts of the sea where there wasn't good deposition happening regularly, and that we just got particularly lucky at this site in Wiltshire, and that they then moved far more. It was far more common for them to migrate in order to occupy areas where there was they were likely to be buried during the lower Cretaceous. So I think a lot of the sort of claims that have been made in the past have been based on lack of evidence, as opposed to being able to make a sort of informed statements on the evidence that we have. It's been more being based on the fact that we don't have any evidence from anywhere else. You see what I mean?
Thank you very much. Well, if I could just use my chair's privilege to ask one final question, perhaps, inspired by John's earlier. What makes this site so very special? Because mud slides down slopes under the sea all the time, and storms move sediment around. Was it a particularly thick covering of mud? I imagine that some of the mobile members of this assemblage could have escaped from burial. You'd imagine the starfish could escape from some sort of burial. So, do you have a sense of how deep this cover was, or whether it was accompanied, by nasty chemical conditions on the seabed or something? What was going on? What's the kill mechanism?
So I think it appears to be burial experiments on modern starfish. Most starfish can only dig their way out of about 5 centimeters of mud. So using that as a sort of ballpark, and we're actually able to see the thickness of the bed at the site. It is about 5 centimeters. That's not to say it probably was a lot thicker coverage, but it's just been compressed down and the fluids have gone out, and so you've lost a lot of that initial depth of burial. So yes, it's probably thick burial. It's thick burial by mud that is fine, which will keep everything together. It will probably become anoxic very quickly. The clay is very dark, so it's obviously anoxic there, and you can see where it's oxidized, where meteoric water has got in through cracks and fissures and things like that. So yes, it was anoxic mud, and then but the seawater itself was probably oxic, and as soon as that burial bed finished, there's good evidence that the crinoids, particularly the Comanches, came back into the area, started procreating furiously, and then they were buried again in a very quick result. And then very soon after that, we had maybe 5 meters of sort of oolitic, sort of limey sands just coming in. You can see all the cross bedding very nicely as it's prograded across the top of the whole thing and buried it under several meters of rock. And that's why it's being preserved. And it's just the perfect quarry that the people digging out the rock only want that nice sort of sandy stuff, and they stop at this horizon where all these echinoderms have come out, and people have just gone down and looked at the right time. It's one of those sort of lots of chances all coming together.
Yeah, a happy series of events indeed.
Yeah, well, very unhappy for them, but great for us. Yeah.
We do have one final comment coming in, so I'll just address that. I can see parallels, taphonomically speaking, with the broadly contemporaneous Bradford clay crinoid fauna of Wiltshire. Do you think that's the case?
Yes and no. So Bradford is a hard ground, and most of the crinoids, all the crinoids that you find there are actually disarticulated. The only articulated bits you find are really the holdfasts and sort of lower bits of the stem, which were obviously cemented together. And all the other bits of crinoids that you find there are like the very large calyxes and all the arms. They're all disarticulated within the mud, and so the similarity is that you have this mud flow burying things very rapidly, but nothing seems to have been preserved whole there, whereas they have been at this site, this other Wiltshire site. I nearly said the name then.
No, we would have forgotten this immediately, of course, closed our ears innocently. Right, well, before we thank Tim once again, can I just give a quick advertisement for the next lecture from the section, which will be on the 8th of February, and that will be from Dr. Phil Bird from the University of Leicester, talking about something quite different, the circular economy and mineral resources. But for now, it just remains to thank Tim again for a really wonderful talk. So from those of us in the room and at home, big round of applause, please. And to everyone who's attended the meeting, thank you for coming. See you here again on the 8th of Feb, and have a good evening.
Thank you.